All right, let's start. So, thank you, Seraphim. It's good to see so many familiar faces out there. We've sort of been hearing for, I guess, a couple of talks now about this problem of enormous data sets, or the promise of enormous data sets, depending on who you ask. And I want to talk about some work that we've been doing recently to try to get a handle on how to compute with huge amounts of genomic data. So first, I'm going to go through, and I apologize to everyone, which I guess at this point is everyone for whom this is a review, but I'm going to talk about how much information is really coming and why this is both a good thing and presents challenges. And then I'm going to give you a bit of a way of approaching it we've come up with, some example algorithms to show that this actually works, and where we're trying to take this now. So let's start with some ancient history. This was sequence analysis just 40 years ago, reading off these very short things, and then not long after is the Human Genome Project, and of course the 1,000 Genomes Project, as we heard yesterday, because of the technological advances, that actually far exceeded 1,000, and we're well beyond it now. And I put up Steve Quake there because that was about the point where he was able to do what they did in the whole Human Genome Project in about 3 weeks. And algorithms have sort of come with this. First, you had the basic local sequence aligners, and then finally there was enough data that, okay, maybe we can't just Smith-Waterman everything, so we have BLAST. And they've come a bit since then. And so I want to talk about what's driving this. And it's really technology. There's all of these technologies, put money into it, and the net result is that what 15 years ago was a $1 billion government scale project, I can now do as a single bench scientist at the lab for just barely more than the amount of money I need to ask permission to spend. And there's plenty of things on the horizon, not these ones necessarily in particular, but there certainly are new technologies coming, so there's reason to believe this is going to improve. And it's been getting cheaper. We've seen similar plots like this. What I want to point out is that here it's getting cheaper by an order of magnitude a year. To put that in terms of computing, which is a little more familiar, that's like going from this Osborne executive to an iPhone in 3 years. Well, I guess at this point, we've all seen this plot. This is the amount of sequencing and computing, both on a log scale, and as you can see, they have quite different slopes. And even in 2011, people were saying, well, our analysis just isn't going to scale. In fact, we're there. It's already a problem. Even a year and a half ago, it cost more to analyze a genome than to sequence it. And what I tend to notice is that in a lot of these articles, there's some vague reference to, we're going to have to turn to cloud computing. And the problem with this is that cloud computing buys you a fixed multiple. You're still constrained by essentially Moore's law. And so the gist, to really distill it, and if you can take one thing from this, it's this plot. When you divide the speed of computing by the speed of sequencing, we're getting slower. We sort of assume, okay, we'll have more data, but we'll also have faster computers. And the trends do not support this. Our ability to analyze future data is actually getting worse and worse. But there are some current variants of BLAST that are constant multiples faster. There are a couple of vendors' booths that have quite impressive improvements. But these don't change the fundamental scaling law. And which is really that anything that's linear, meaning any algorithm you do that looks at the full data set of a genomic library, or even some constant fraction thereof, is getting slower and slower. Sorry? And so how do we get around this problem of, if we look at all our data, we can't do this in finite time? Well, the trick is, of course, to not use the full data set. Specifically, to leverage genomic redundancy, that when we sequence multiple organisms that are related, there's a ton of overlap. We're not actually getting a full genome worth of new data. In some sense, if I was to sequence Seraphim and do some computation on his genome, and then I wanted to do the same computation on the genomes of every other person in this room, everyone is so closely related. In some sense, by doing that computation, I've already done 99.9% of the work to do the same computation on everyone else. So instead of taking 300 times the time, it should take something like 1.1 times the time. And really what we want to do, because this is the fundamental bound, is not computation that scales with the size of the database, but computation that scales with the entropy of the database, the total non-redundant information. And to sort of drive this intuitively, when we ask biological questions, typically the sort of thing we're asking is what regions of the genome are similar between people or to our reference, and what regions are not. Not so much exactly what the sequence is, but really what's like what. And this is a much sparser problem. And so there should be some intuition that we're really asking questions of some very sparse subset of the full genomic library. Now, this is not trivial, and the reason for this, well, this is compression. So here, this gray block, let's call this a genome. And suppose these two regions are similar. So in classic compression, what you're going to do is you're just going to replace one with a pointer to the other. And the problem with this is that we compress on some optimal compression metric, which isn't necessarily the sparsification that we're asking about when we want to do some algorithm like BLAST. And so what we have to resort to is unpacking this data first and then analyzing it. This has actually been done quite beautifully in the exact matching case with the FM index, which I really encourage everyone to look up. It's a beautiful paper. But we want to ask this question about inexact matches, where we have all of these, we want to say, let's find everything that's kind of like this. And that actually gets much trickier. So let me show you how we do it. To get a handle on computing, we thought, let's go after the one algorithm that we're sure that everyone knows about for approaching computing, BLAST. I mean, it's to the point where I don't even probably need to tell anyone in this room what it stands for. And the way we're going to do this is first, we're going to compress the data down to some compressed library, and then show how we can search and do something that's basically the same as BLAST on this compressed data in time proportionate to it. So to remind those of you for whom it's been a while, this is basically how BLAST works. You take some user query, you parse it up, you find the exact matches in the genomic database, and then you expand these matches and say, okay, how good are these expansions, score them, and return. And what we do is we take this database and we do a single compression phase first. So we have some compressed database. And then on this compressed database, whenever you have a query, then we run this search phase, which actually gives BLAST results right back. For the compression, what we do is basically the first most naive thing we could think of for doing compression. And I am sure there are better ways to do this. Hopefully, there are people in the audience who will tell me afterwards better ways to do this compression. But what we basically did is we said, all right, let's look at regions that only have a few edits between them, and then replace with pointers plus a few edits here and there. So we take long sequences and say, okay, well, in these 5 points, that long sequence is different. And that's sort of it. So now the search phase. So here what we do is we say, all right, we have these compressed things, so let's relax BLAST a little bit and make it a little more permissive. Search through the database, we get this small set of very permissive hits, and then we expand each of those without having to decompress the whole thing and say which of those actually link to real BLAST hits. we also got asked if we could do the same thing for BLAT, since that's another indexing method that some of you may have heard of, or probably have. And what I want to drive in with this sort of coarsely made slide is that they're really the same thing. We take the same database, and instead now we just do a coarse BLAT, and then use BLAT's fine search to refine the results. And so from there, we got our results. And now we have the problem, well, does this actually work faster? And there's a bit of subtlety in this question, because really what we're asking is, how are we going to perform on data we don't have yet in response to queries that we don't know yet? So we have to guess. And for example data sets, so we sort of use the intuition that a lot of the effort up until recently has been to fill out the tree of life with sequencing, and now we're working to fill it back in, to come up with things that are more similar to sequences we've already found. And so we take a database of very similar yeast genomes. We did the fly genomes, and then we also did some simulated data. And I'm going to tell you about the results in the yeast only in this talk. And then we need example queries. What do people search for? Because the entire game here is dependent on the data, the structure in the data and the similarity of it to what we already have. And so you can't really use random searches. And so what we did is we said, all right, probably what people are going to be doing is searching for genes that they've found elsewhere. So what we did is we took random bits of genomes, we mutated them, threw out low complexity things, and then said, okay, on this very large set of putative queries, how do you do? Well, here's how we do. This black line is this is the file size, and these are the raw run times of normal, BLAST, and BLAT. And then the blue is how long the entire thing takes. And the red is the coarse phase only. And what we see is not only do we do dramatically faster, but it gets better with more data. We actually really change the scaling. Now, there are some trade-offs here. There's a slight loss of accuracy and specificity. And here we can tune this. Because here, when we do the compression, we have a choice of how strongly do we want to compress. And there we're really trading off compression and speed versus some amount of accuracy. But it's in fact not a huge amount of accuracy. We about 99%. And the ones we miss are primarily these marginal cases that typically are not quite statistically significant anyway. In search, you have the same thing. You can say, okay, how permissive do you want to be on this coarse search? Because you increase your chances. And the reason there are these trade-offs, the reason we can't do it perfectly, is that I've been using this notion of sequence similarity, but I really mean two things. For compression, what I mean is absolute similarity. When you have two sequences, what fraction of them are similar? But for BLAST, we're not asking that. We're asking, what's the probability of similarity? So if we have a run of 1,000 bases that's 50% similar, that's a great BLAST hit. But it's an absolutely atrocious you can't get any compression from that whatsoever. And this is really a question of the metrics. And so, suppose we have a query, and there's some target we'd like to find, and it's BLAST similar. But instead what we do is we compress it to this consensus, and now we're going to search and find this consensus. And so we're really conflating these two metrics, and so we don't even have the triangle inequality, but we don't have to relax it that much to be able to get this to work. And so that's sort of it for KBLAST and KBLAT. What we've shown is it runs in time proportional to the non-redundant data. We scale much more favorably. We actually get increasingly advantageous as you get more genomes. And we get over 99% accuracy and 100% specificity. The reason for the 100% specificity is that the final step is, in fact, the exact same algorithm as the ones we're comparing to, and so we're never going to get something that it's not going to call. So let me talk a bit about where this is going in my remaining 5 or so minutes. The first thing I'd like to do is give a bit of a preview for a talk this afternoon given by Noah Daniels on protein blast. The general spoiler is it works. But I encourage everyone to go to this talk because the details of it are actually very interesting. It presents a lot of different challenges working in protein space versus genome space. And ultimately, while this sort of single-step compression works for now, in order to really get a logarithmic speed up, we have to go hierarchical. This isn't going to last. A scheme like this is not stable completely to future data. And we'd like to also ask, what are some other questions? So what other things can we do? Because there are plenty of things that aren't BLAST you can do in genomes. You can run hidden Markov models. You can look for, well, for proteins, you do threading. You can look for signatures of evolution and so forth. And we're moving into that right now. And then ultimately, once we've compressed it, there should be some biological inference in this compression. You can see this because under the sort of maximum parsimony hypothesis, really what it's saying is that the optimal compression scheme is exactly the phylogeny, because that's where you put in the minimum amount of new information. So that's sort of it. I want to thank very much the incredibly talented Po Ru Lo, who did the majority of the coding for this, Bonnie Berger, our funding sources, and of course the travel fellowship from the NSF. So to recap, sequencing is fast compared to computers, and this main point that linear algorithms on the present data set are getting slower, effectively. Showing you a couple of a couple of new algorithms for applying BLAST and BLAT to this. And then more generally, the title, Compressive Genomics, is meant to sort of introduce this idea of compressing down our data and looking really at the non-redundant bits. Software is freely available. This is the citation, and I'm happy to take questions. Do you want to moderate? No.
Yep, the talk that shows in 3 lines. Use the mic. Sorry? Just use the mic. Oh, okay.
Slides are not that long.
So the plot that you showed with 3 lines suggests just a constant factor improvement, to speed up. But you said that this scales with non-redundant data.
It was a little bit of an illusion. So that was a little bit of an optical illusion. It actually is plateauing. You tend to see this more when we did the simulated data. But that is slightly sublinear. It's just at the slope that it's at, it's very difficult to see in the presentation.
Okay.
Hey, Michael. Very clear presentation. Thank you. So there's the other side of the coin as well. The BLAST model is, although it's very valid, it's changing a little bit because the query is not a single sequence anymore, right?
Right.
It's just a lot of stuff queried at the same time. Have you guys been thinking in these lines how to combine the compression programs for the input with the compression programs for the database itself?
So I guess I should probably repeat the question quickly. This was asking whether we're compressing inputs as well, because if, say, you're blasting an entire Illumina lane, often those things are very similar to each other. The answer is yes, we are doing that, and in fact, your former student is currently about to submit a paper on this.
Good to know.
How easy or how difficult is it to update these compressed databases when you have more and more sequences coming in?
Right now, the way we have it working, we have to recompress the entire thing. But ultimately, it's actually a very similar step to sort of iteratively add things on. That's an area that we're looking at, because this compression scheme right now is not incredibly robust to future additions.
You could imagine that you could constantly having new sequences coming in, and that iterative process would be unfeasible.
Yeah. I mean, hopefully there one can distribute this by actually making the computer attached to each sequencer sort of do the marginal work there, but something like this. So this is really a concern there too.